The overall goal of this procedure is to culture an enriched population of dissociated primary sensory neurons from chicken embryos. This is accomplished by collecting intact dorsal root ganglia from the embryonic day 7 to 10 chicken embryos into a conical tube and subsequently breaking them up into a dissociated cell suspension. As a second step, dissociated DRG cells are plated on a culture dish for incubation. Afterward, the culture dish is gently rinsed to preferentially dislodge the neurons. The final step includes plating the dissociated sensory neurons on the well-defined substrata, such as acid-washed and baked cover slips pre-coated with high or low concentrations of laminin-1. Immunocytochemistry is used to show the presence of NCAM and activated beta-1 integrins in the cell bodies, neurites, and growth cones of the dissociated cultured embryonic chick sensory neurons.
This method is a powerful tool to elucidate numerous aspects of neurobiology, including mechanisms of axon guidance, growth cone cytoskeletal dynamics, as well as cell surface composition and regulation.
This technique allows researchers to rapidly obtain the cultures highly enriched with dissociated sensory neurons, which contain very few glial cells. This is distinct from the intact dorsal root ganglion cultures that contain both neurons and non-neuronal cells.
Furthermore, minor variations of this protocol should allow high yield of neuronal cultures from sources other than dorsal root ganglia, including embryonic forebrain.
In a laminar flow hood, crack a staged fertile white leghorn chick egg and place its contents into a 100 mm Petri dish.